Hello everyone and welcome. We are team VIJAYA Lakshmi Pandit and we are excited to present our communication skill project on a very engaging topic that is brain's biggest secret, which is narrated by Melanie E. Pefa. I am Rituraj and I am looking forward to exploring the fascinating world of our brains with you all.
Hi everyone, I'm Annu. I'm particularly interested in how the concept in this video relate to our everyday learning experience. Let's dive in.
Hey everyone, I am Anoop and I am eager to share our interpretations of this insightful video and hopefully make some of these complex ideas more accessible. Hello viewers, Devendra here. I am keen to see how effectively we can communicate this key message of brain biggest secret through our performance.
Hi, I am Denish. I am excited to be a part of this project and to bring Melanie Epipher's narration to life in our own way.
Hi everyone, Saurav here. I hope you enjoyed our video on this thought-provoking video about the brain's hidden potential.
So get ready to unlock some of the brain's biggest secret with us. So let's get started. The 19th century was a period of both intellectual ferment and deeply intensive scientific dogma. As scientists across Europe and beyond emerged an ambitious quest to decipher the fundamental laws of nature, the human body remained one of the most captivating and enigmatic frontiers. Among the many puzzles that intrigued researchers, the nervous system stood out as particularly complex and challenging. This intricate network, responsible for orchestrating everything from the simplest reflex to the most sophisticated cognitive function, seem to defy easy explanation. Within the scientific community, a particular concept gained widespread acceptance, shaping the direction of research and understanding for a significant portion of the century, the reticular theory. This theory proposed a model of the nervous system as a single continuous reticulum, a vast web where individual cells believed to be physically connected, forming a seamless and uninterrupted network.
The reticular theory, championed by many of the era's most influential scientists and anatomists, offered an elegant and seemingly comprehensive explanation for how the nervous system functions. It painted a picture of the nervous system as a unified whole, a single entity in which individual cells most lost their individuality, merging into a continuous, interconnected structure. To illustrate this concept, prominent scientists often drew open, vivid, and relatable analogies. The nervous system was likened to a vast and complex river network where water flows uninterrupted from the smallest tributary to the mightiest river and finally to the sea. Or it was compared to an intricately woven tapestry where individual threads intertwine and interconnected to create a single, unbroken design. This analogy served to reinforce the idea that the nervous system operated as a single, unified system with signals propagating throughout the network in a continuous, seamless and fashion, much like the flow of water in a river or the interconnectedness of thread is a tapestry.
However, this prevailing view, despite its widespread acceptance, did not go unchallenged. In Spain, a young and ambitious scientist named Santiago Ramon y Cajal began to develop a dissenting perspective, working often in relatively modest conditions and with limited resources. Cajal dedicated himself to the meticulous observations of nervous tissue under the microscope. His approach was characterized by a relentless pursuit of detail, a commitment to empirical evidence, and a willingness to question even the most deeply entrenched scientific dogma. as he peered through the lens of this microscope, carefully examining the intricate structure of the brain and spinal cord. Casal's observations began to diverge significantly from the prevailing reticular model. He saw not a continuous interconnected web, but rather a collection of individual cells, each with its own distinct structure, boundaries, and processes. Casal's unique perspective and his ability to challenge the established view were profoundly influenced by his background and training. In his youth, he had received extensive artistic training, developing a keen eye for a detail, a deep appreciation for form and structure, and an ability to translate three-dimensional objects into two-dimensional surface. He approached the study of the nervous system with this artist's sensibility, meticulously capturing the intricate forms and the subtle nuances of the cells. He observed, for Casal, drawing was not merely a means of recording observations, it was an indispensable tool for scientific discovery. The act of translating the microscopic world onto paper forced him to analyze and interpret what he saw, to discern patterns and relationships that might otherwise have gone unnoticed. Through his drawings, Casal began to develop a new and revolutionary vision of the nervous system's organization, one that directly contradicted the prevailing reticular theory.
When Castle presented his groundbreaking findings and his radical new interpretation of the nervous system to the wider scientific community, his challenge to the reticular theory ignited a passionate and often contentious debate. The established scientists, many of whom have dedicated their entire careers to the reticular model, were often deeply resistant to the accepting his revolutionary ideas. Accustomed to the elegance and apparent simplicity of the continuous network concept, they struggle to reconcile Casal's meticulous observations with their existing theoretical framework. A wave of skepticism and doubt was over Casal's claims. Questions arose about the validity of his techniques, the potential for artifacts in his staining procedures, and the accuracy of his interpretation of the microscopy images. Were the gaps that Casal observed between cells merely illusions, imperfections of their preparation processes, rather than real structures, features of the nervous system?
The clash between Caesar's groundbreaking work and the prevailing reticular theory represented far more than just a simple scientific disagreement or a debate over the interpretation of data. It highlighted a fundamental tension inherent in the process of scientific discovery. It in conflict between established knowledge and emergence of revolutionary new ideas. The scientific community stood at a critical crossroads, forced to confront the unsettling possibility that a long held and widely accepted belief might be fundamentally flawed. This pivoted moment marked the beginning of a paradigm shift, a profound transformation in the understanding of nervous system that would ultimately reshape the course of nuisance and pave the way for our modern understanding of the brain.
Now, the story of the neuron doctrine, the revolutionary idea that the nervous system is composed of individual cells, is not solely a tale of observation and debate. It is also a story of a crucial technological advancement, a serendipitous discovery that provided scientists with an unprecedented window into the microscopic world. This breakthrough came in the form of a new staining technique, a method that would selectively illuminate the intricate structure of nerve cells with breathtaking clarity. The architect of this technique was Camillo Golgi. Camillo Golgi, he was an Italian physician and he was an extraordinary scientist working in Pavia. Golgi, though a prominent of the reticular theory, inadvertently provided Castle with the very tool he needed to dismantle that theory.
Golgi's primary research interest lays elsewhere. He was focused on other histological studies. In his pursuit of better ways to visualize nervous tissue, Golgi experimented with various metallic impregnation methods. It was in this context, almost by accident, that he stumbled upon what would become known as the region nera or the black reaction. This technique involved treating brain tissue with potassium dichromate followed by silver nitrate. The result was astonishing, a selective staining process that, for reasons not fully understood at the time, impregnated only a limited number of neurons in their entirety from the cell body to the furthest reaches of the axons and dendrites. This was a radical departure from previous staining methods, which tended to stain all cells indiscriminately, creating a confusing, impenetrable mess under the microscope.
The impact of Golgi's staining technique was immediate and profound. For the first time, scientists could visualize the complete morphology of individual neurons in explicit detail. The intricate branches of dendrites, the long slender axons, and the cell bodies all stood out with startling clarity against the background tissue. Initially, even Golgi himself interpreted these images through the lens of the reticular theory. However, when Castle adopted Golgi's method, the results fueled his conviction in the neuron doctrine. For Castle, the Golgi stain was nothing short of a revelation. It was as if a veil has been lifted, revealing the hidden architecture of the nervous system with breathtaking clarity. Through the microscope, he could now trace the intricate pathways of individual neurons, map their connections, and observe the spaces that separated them. The clarity and detail provided by the stain offered irrefutable evidence for what Cazal had long suspected. The nervous system was not a continuous reticulum, but a network of discrete cells. Kazan dedicated himself to meticulously
documenting his observations, creating a series of detailed drawings that captured the beauty and complexity of strain neurons. He traced the paths of axons, mapped the branching patterns of dendrites, and carefully noted the points of contact between cells. In his drawings and accompanying descriptions, Castle articulated a new vision of nervous system, one based on the principle of individual cellular units. He proposed that nerve signals traveled us in a specific directions from the dendrites through the cell body and along the axon to the next neuron. This dynamic polarization, as he called it, further supported the idea of distinct cells communicating with each other.
Tissue in general became the key that unlocked one of the biggest secrets of brain. It provided the visual evidence needed to solidify the neuron doctrine, shifting the paradigm in neuroscience. Through Golgi and Kazal initiated disagreed in their interpretation. The power of the technique was undeniable. It transformed the study of the nervous system, allowing scientists to move beyond speculations and into the era of detailed observation and analysis. The debate ignited by the stain pushed the field forward, ultimately leading to a more accurate understanding of the brain's fundamental structure.
The groundbreaking work of Castle, fueled by the clarity of Golgi's stunning technique, began to trickle through the scientific community. As more researchers adopted the new method, of a wave of fresh observations and corroborating evidence emerged, scientists in different corners of the world started to witness the same cellular individuality that Castle had so passionately described. The idea that the nervous system was composed of discrete units, the neuron doctrine, slowly began to gain traction, challenging the long held assumptions of the reticular theory. This period was marked by a flurry of activity. New studies were published, debates read in scientific societies, and very foundations of neurosciences were being re-examined.
The scientific community, however, is rarely monolithic. While the evidence for the neuron doctrine mounted, skepticism and resistance persisted. Some prominent figure deeply invested in the reticular theory or hesitated to abandon established ideas, remained unconvinced. They questioned the interpretation of the new findings, suggesting alternative explanation or clinging to the belief that the nervous system, as its core, was a continuous network. This reluctance created a tension within the field, a push and a pull between the old guard and the proponents of the new paradigm. The debate became a driving force, pushing scientists to refine their techniques, rather more data and strengthen their arguments.
The growing body of evidence supporting the neuron doctrine came from diverse sources. Researchers began to identify different types of neurons, each with unique shapes and functions, further reinforcing the idea of cellular specialization within the nervous system. Studies of neural development revealed how individual neurons grow and connect, providing insights into further formation of neural circuits. Pathological investigations examining diseased tissues showed how damage to specific neurons could lead to specific neurological deficits. This convergence of evidence from multiple lines of inquiry gradually eroded the resistance to the neuron doctrine, paving the way for its eventual acceptance as the cornerstone of modern neuroscience. The culmination of this scientific revolution arrived with the recognition of the key figures who had shaped it. In 1906, Camillo Golgi and Santiago Ramon y Cajal were jointly awarded the Nobel Prize in Physiology or Medicine. This prestigious award acknowledged the immense significance of their contributions to our understanding of the nervous system's structure. However, the Nobel ceremony also highlighted the lingering tensions between the two scientists. Despite sharing the prize, Golgi and Cazale used their Nobel lectures to reiterate their differing interpretations of the nervous system, showcasing the enduring power of their respective viewpoints.
Golgi, even in his noble culture, continued to defend the reticular theory, emphasizing the interconnectedness of nerve cells. Castle, on the other hand, used the occasion to further elaborate on the neuron doctrine, presenting compelling evidence for the individuality of neurons and their role in neural communication. This juxtaposition of opposing viewpoints, presented on the world's most prestigious scientific stage, created a dramatic irony. It underscored the complexity of scientific progress, where even in moments of triumph, old ideas can persist alongside new discoveries.
The legacy of Siegel's neuron doctrine extended beyond the 20th century. It laid the essential foundation of our modern understanding of the brain, influencing generations of the neuroscientists and shaping the direction of research of this day. His work provided the framework for exploring how neurons communicate through synapses, how circuits passes information, and how the brain limbs remembers and gives rise to consciousness. Modern techniques from the electron microscopy to fluorescent labeling have provided even more detailed and dynamic view of neurons and their interactions, confirming and expanding upon Siegel's original insights. The journey to understand the brain's biggest secret, the dance of individual neurons, continues, but its origin lie firmly in the groundbreaking work of Siegel and the power of the stain that illuminated the path.
The story we have explored isn't just about what we have discovered, it's about how much remains to be unveiled. The shift to the neuron doctrine was a monumental leap, but it also revealed the sheer complexity of what we are trying to understand.
Consider Kaza's drawing. They are beautiful and precise, yet they capture only a static moment in a dynamic system. What secret lies in the living dance of neurons, in the fleeting connection that shape our thoughts and memories?
Golgi's stain, for all its power, only illuminates a fraction of the cellular landscape. What about the vast network of glial cells, the supporting cast of the brain, whose roles we are only beginning to appreciate? The journey from seeing the brain as a continuous web to understanding it as a collection of individual units was profound. But what new perspectives await us? What future revolutions will shape our understanding of this intricate organ?
The neuron, that fundamental building block, is the both simply and astonishingly complex. Each one a universe in itself, and together they create the most complex structure known to us.
So, as we close this chapter, let the exploration continues in your own mind. The brain's biggest secret is not just something to be observed, it's something to be pondered, to be marveled at, a mystery that invites us all to become lifelong students of the mind.